Hey guys, it's Mr. Barnett. Just going to be going over the biology EOC review we had last week in class. We weren't able to finish, so please make sure to take notes on this and make sure to listen and pay attention, and hopefully this will be of some use for you guys for the EOC. So the first question, number one, a team of ecologists observed feeding patterns of several populations in the desert. The energy pyramid shown below depicts the feeding patterns the ecologists observed. So this is what we call an energy diagram, and it shows the flow of energy in an ecosystem and how energy travels from the primary producers up to the apex predators. And we call each of these columns on the energy pyramid a trophic level. So if you see over here, this is the fourth trophic level, the third trophic level, the second trophic level, and the first trophic level. The trophic level highest up on the energy pyramid is lowest in energy, and the trophic level furthest down on the pyramid is highest in energy. And if we take a look here, that makes sense because once again, at the bottom of our energy pyramid, these are our primary producers, also known as photosynthetic organisms such as plants and photosynthetic bacteria, things that undergo photosynthesis and produce glucose and oxygen. That glucose is a form of chemical energy that can then be consumed by the primary consumers, which is one level up here. But what you really need to know is that energy travels upward in this pyramid. And at each trophic level, there is less and less energy. So energy travels upwards, and as we get to each level, there's less and less energy. And that's because we're losing energy due to the organisms having to live in their environment, walk around, move around, reproduce, and also just losing energy in the form of heat to the actual external environment they're present in. But we once again need to know that this first trophic level, the level at the bottom of the pyramid, is highest in energy, and the trophic level at the top of this pyramid is lowest in energy. Excuse me. So, when taking a look at this, let's read this. Which of the following best explains the difference in the amount of available energy in the trophic levels of the desert ecosystem? Question A says there is less energy available in the producers because their tissues are less dense than those at higher trophic levels. That doesn't make any sense because, once again, the primary producers are at the bottom. Apex predators are at the top. And we know that the primary producers are highest in energy, so we can eliminate that one. That doesn't make any sense. B says there is more energy available in the second trophic level because less energy is needed for hunting compared to the higher trophic levels. That really, we can't get any of that information from just looking at the energy pyramid, so B doesn't make much sense. C, there is more available energy in the birds of prey because they have greater muscle mass for storing energy than organisms lower trophic levels have. Once again, C doesn't make sense because the higher up we go on this pyramid, the less energy is available. D, there is less available energy in the fourth trophic level because of loss of energy through metabolism in each of the lower trophic levels. This is the one we would want to choose because at each of these levels, there is less and less of these organisms, and they are losing their energy just due to their metabolism and working around and walking around and reproducing. So D would make the most sense. But the easiest way to remember this is energy decreases as we go up each trophic level. The level at the bottom of the pyramid is highest energy, and the level at the top of the pyramid is lowest energy. And we call the organisms at the bottom of this pyramid primary producers. We call the organisms at the top of this pyramid apex predators. So moving on. Water is essential for life. Its special properties make water the single most important molecule in plant life. Which of the following properties of water enable it to move from the roots to the leaves of plants? So water can also be written as H2O. And water is a unique molecule because H2O is polar, and that's due to differences in the electronegativity present on the oxygen atoms and hydrogen atoms that make up H2O, which makes the electrical charge present on H2O unevenly distributed. And H2O, water, is also cohesive / adhesive. And what that means is water, H2O molecules like to stick to themselves because polar attracts polar, but H2O molecules also like to stick to other things, such as the xylem, the vascular system present in plants. So H2O sticks to things very well. That's why if you throw water on a wall, it doesn't just immediately start sliding down. There'll be a couple droplets that stick on the wall, a couple droplets that fall down, a couple droplets that disperse, but H2O, water, is cohesive and adhesive. Water also has a very high specific heat. And what this means is that water H2O, has to absorb a lot of energy before we see a change in its temperature. And if water has to absorb a lot of energy in order to see a change in its temperature, that means that large bodies of water usually remain at constant temperatures. That's why if you go into the ocean on a really hot day, the ocean water still might feel really cold even though the atmospheric temperature is really high. That's because the water present in the ocean hasn't absorbed enough energy yet for the large body of water to actually have its overall temperature change. So we're still going to associate that large body of water as being cold, even if the atmospheric temperature is really high. And that's because H2O has a high specific heat. Water has to absorb a lot of energy before we see a change in its overall temperature. So water is able to moderate itself very well, its temperature very well, and that's because it has a high specific heat. We also need to know that water, H2O, is an excellent solvent. And what a solvent is something that can dissolve things. So the solute is the thing that is dissolved, and the solvent is the thing that dissolves the solute. But water is the best biological solvent out there. And that's due to it being polar and having dipole moments. You'll learn more about this when you go into chemistry. But for right now, just keep in the back of your mind that water is the best biological solvent, especially for the EOC. Now, this question though, even though technically each of these is explaining a property of water, what property of water would explain why it's able to move from the roots to the leaves of plants? So why can water move from the roots to the leaves of plants? Which of these properties would allow it to do that? Well, let's think of it this way. If water has to start from the roots, which are down here, and get to the leaves, which are up here, it would have to travel upwards against gravity, because gravity is going to be pushing the actual water back down. So there would have to be some mechanism that allows for the water to travel upwards against gravity, or else the water would never be able to go from the roots to the leaves, and the leaves require water in order for photosynthesis to occur. But the reason the water can travel through the vascular tissue known as xylem present in plants is because H2O has this property right here. It's cohesive and adhesive. H2O is able to stick to unlike materials very well, which would allow it to travel from the roots up to the leaves. So we can eliminate water expands as it freezes. That makes no sense for this question. Water expanding as it freezes doesn't explain how water goes from roots to the leaves. Water is an excellent solvent. That doesn't make any sense either for the situation at hand, because just because water can dissolve things really well doesn't make sense of why it's able to move from the roots to the leaves. So we can eliminate B. Water exhibits cohesive behavior. Now that's what we're looking for. That is the correct answer. But let's just look at D. Water is able to moderate temperatures. That's true. Water does have a very high specific heat, but does that explain why it's able to move from the roots to the leaves of plants? No. So we can eliminate that one, and it leaves us with C. Moving on to number 3. An osmosis investigation was conducted using chicken eggs to represent cells with semi-permeable membranes. The mass of each egg was measured to determine how much water to fuse into or out of the eggs. The eggs were first soaked in vinegar to dissolve the shell. Each egg was then placed in one of 3 different solutions for 24 hours. The table below shows the results of the investigation. So, you're going to have to be able to analyze graphs. And we've been doing this all year. But when you're given a graph on a test, most likely the question you're going to be asked is going to relate to the graph. Actually, I'm just going to say it. 99.9 to 100% of the time, the answer is present in the graph. So if you can eliminate any answer choices that don't relate to the graph or the graph doesn't explain, you're in a better position. So let's take a look at what the question's actually asking us now. Based on this experiment, which of the following should be inferred about cells with semi-permeable membranes? So we want to look at the graph. Question A states substances other than water may also cross the cell membrane. That's technically true. Carbon dioxide can cross the cellular membrane passively. Oxygen can cross the semi-permeable membrane present in the cell membrane passively. But this graph isn't explaining any of those molecules. It's only talking about water, vinegar, corn syrup, and distilled water. So we can eliminate A right off the bat because this graph doesn't explain anything about other molecules besides water crossing the semi-permeable membrane present on the eggs. Question or answer B states substances other than water may block pores in the cell membrane. Once again, the graph doesn't tell us anything about the pores being blocked. So we can eliminate answer B. Answer C states water enters the cell when placed in environments of high water concentration. So keep in mind, when we're talking about osmosis, osmosis is when water goes from a high concentration to a low concentration without the use of energy. So water goes from a high concentration to a low concentration without energy. And it is a form of passive transport, meaning that it utilizes a semi-permeable membrane along with no energy. So there's no input of energy for osmosis to occur. H2O molecules will just go from a higher concentration to a lower concentration across a semi-permeable membrane without the use of energy. So keep that in mind whenever you hear osmosis in a question. But we can even look at this graph and we see that water enters the cell when placed in environments of high water concentration. So if we put the egg in an environment that has 100% water concentration, you notice that the mass of the egg before was 64.5 grams. And afterwards, it was 105.3 grams. So where did those extra grams come from? That came from water entering the overall egg. So that means that C is our correct answer. And we can see from the graph, before we placed it in the water, it was 64.5 grams. But then after we placed it in water, due to osmosis, the water going from a higher concentration to a lower concentration inside of the egg became 105.3 grams. So that means that C is explaining this perfectly. Water enters the cell when placed in environments of high water concentration. D, don't get tricked with D, because D states water leaves the cell when it's placed in environments with a low concentration of solutes. Remember, if you have a low concentration of solutes, you have a high concentration of water. And if you have a high concentration of solutes, you have a low concentration of water. And once again, water goes from high concentration of H2O to a low concentration of H2O when talking about osmosis. So, moving on. 4. One of the accepted scientific theories describing the origin of life on Earth is known as chemical evolution. According to this theory, which of the following events would need to occur first for life to evolve? So, this would be a trickier question on the EOC, but chemical evolution basically states that before life could evolve on Earth, there had to be genetic material. Because remember, cell theory states all living cells require DNA RNA. So chemical evolution states that before life on Earth could occur, there had to be DNA or RNA. But before we could have DNA or RNA, they're asking what had to happen first. So before DNA or RNA could occur, you would have had to have had organic molecules, because DNA and RNA are made of organic compounds / molecules. So before we could even have DNA and RNA, we had to synthesize organic molecules, which would be C. So to keep this in mind, think of it this way. Chemical evolution was, first thing that had to happen was organic molecules had to occur / compounds, and then 2, second thing that would have had to have occurred would be the synthesis of DNA / RNA, and then from that we could have our primitive unicellular prokaryote occur. Now, one other thing I will note is that when we talk about life on Earth, the origin of life on Earth, we need to remember that technically we all share a common ancestor, and that common ancestor is a heterotrophic prokaryote. This was the very first organism on Earth. And then the heterotrophic prokaryote actually underwent evolutionary mechanisms that led to the divergence of autotrophic photosynthetic prokaryotes occurring. And then from these autotrophic photosynthetic prokaryotes, this led to the emergence of our atmosphere, because one of the byproducts of photosynthesis is oxygen. So emergence of atmosphere due to oxygen being released. And then once there was oxygen in our atmosphere, this led to the emergence of primitive unicellular cellular eukaryotes. So keep that in mind also for the EOC, that when we're talking about the origin of life, the very first thing to occur was heterotrophic prokaryotes. And that's there. Then these heterotrophic prokaryotes diverged into autotrophic photosynthetic prokaryotes. Now, the important thing to keep in mind here is the word photosynthetic, because from these autotrophic photosynthetic prokaryotes, the emergence of our atmosphere occurred due to one of the byproducts of photosynthesis being oxygen. This oxygen being released into our atmosphere led to more complex organisms being able to evolve, such as primitive unicellular eukaryotes. Because remember, some eukaryotes rely on cellular respiration. Any questions on that? So for this one, we would have said synthesis of organic molecules. So moving on. So the next question states hemophilia is a sex-linked, let's highlight that word, sex-linked recessive trait. Which of the following describes the probability of hemophilia in the offspring of a man who does not have hemophilia and a woman who is heterozygous carrier? So remember, heterozygous is when we have one dominant allele, one recessive allele. Homozygous is when we have two recessive alleles or two dominant alleles. And this question says we're dealing with a sex-linked recessive trait. So when you see the word sex-linked, and it's asking you for percentages, that means we're going to be utilizing X and Y and XX. XY is your male, XX is your female. Now, if it's sex-linked and it's linked to the X chromosome, we draw the linked disease allele like this. So if there's a little asterisk above it, that means the diseased allele is present on that X chromosome. If there's not an asterisk present on that X chromosome, this is healthy. Now, for females, they would need both of their X chromosomes to be affected in order for them to be diseased. They just have one. They are what we call carriers. But if both of their X chromosomes are diseased, then they are actually expressing the overall diseased phenotype. Males, on the other hand, just need one of their X chromosomes, because they only have a single X chromosome on their 23rd pair, to be diseased. Because their second chromosome on their 23rd pair is going to be a Y chromosome. And if it's sex-linked on the X chromosome, they only have one X chromosome to give. So if their single X chromosome possesses the diseased allele, they are going to actually express the disease. So this states that we have a man does not have hemophilia. So he would be XY, just like so. And then we have a woman who is a heterozygous carrier, meaning that one of her X chromosomes is going to have the diseased allele on it, and one of her X chromosomes is going to be normal. So we'll draw this out like so. We have the male on top, which I'll write M, and the female on the left. And we just draw this like a normal Punnett square. And then make sure to also carry over the diseased allele. So, if we take a look at this now, the percentage of male offspring is going to be 50%, and the percentage of female offspring would be 50%. So they have a 50% chance to have a female, 50% chance to have a male. They also have a 25% chance to have a diseased child. So there's a 25% chance to have a diseased child. And we can see that because the only child that could be diseased would be this child right here that I have circled, and that's 1 out of 4. Now, if we were talking about the percentage of diseased males, the percentage of having a diseased male would be 1 out of 2, which would be 50%. And that's because when we're just dealing with males, just focused on this side right here, we only have 1 male that could be diseased out of 2, so that would be 1 out of 2, which is a 50% chance that we have a diseased male. But we are talking about the probability that their offspring, so their entire offspring, this entire chart, we don't care whether they're male or female. We care about all their offspring in this question. We want to know the probability that of hemophilia in the offspring. So there is only a 1 out of 4 chance, which is 25%, that one of their offspring, out of all the offspring possible, would have hemophilia, because it's only this male right here that would actually be able to express the diseased phenotype. This female right here is just a carrier. This female in the bottom left is completely healthy, and this male in the bottom right is completely healthy. So we would say C. There's a 25% chance that their sons will have hemophilia. Any question? Moving on. So here we have a sodium-potassium pump. And remember, the sodium-potassium pump is a form of primary active transport. Primary active transport is when you go from a lower concentration to a higher concentration against the gradient across a transport protein utilizing ATP, also known as adenosine triphosphate. And these right here are transport proteins. And remember, proteins are made up of amino acids. These could be any type of particle that I have highlighted, but if we're talking about the sodium-potassium pump, this would be sodium, and this would be potassium. But don't even worry about that. Just read what they have to say and answer the questions. So a cell membrane is composed of a double layer of lipids in which many kinds of proteins are embedded. Many of these proteins act like gates, allowing only certain particles to enter or leave the cell. When a particle has to go against its concentration gradient through one of these gates, energy is supplied by the cell to the correct protein to move the particle through the membrane. Sodium-potassium ion pump is an example of a type of carrier protein that uses a large portion of the cell's energy to move sodium ions through the cell membrane. So I would like to stop here and just review a little bit. Remember, when we're talking about the cell membrane, it's made of these phosphate heads. And these fatty acid tails. And the interior portion of this phospholipid bilayer is hydrophobic, and this exterior portion is hydrophilic, meaning it's water loving. So where I'm coloring in blue, this is hydrophobic, meaning it's water hating, and where I'm coloring in red, these outside phosphate heads, they are hydrophilic, meaning they're water loving. And we call this the phospholipid bilayer, and the phospholipid bilayer makes up the cell membrane that surrounds the entire cell. It's composed of those fatty acid tails that are hydrophobic and those phosphate heads that are hydrophilic. But let's actually look at the questions they're asking. So, the sodium-potassium ion pump found in some cell membranes is made of which of the following basic structural components? So, the actual pumps are made of amino acids, because remember, the two big things we need to know that proteins make up. Proteins, once again, are made of amino acids. And proteins are used to make up enzymes and also transport. So protein transporters are made of proteins, and proteins are made of amino acid, and enzymes are made of proteins. And once again, proteins are made up of the monomers amino acids. So if you ever see the word transporter, or you ever see the word enzyme or carrier channel or anything like that, you're going to think of protein. If you ever hear the word protein, you're going to think of the word amino acid. If you ever hear the word enzyme and it's asking what enzymes are made of, you're also going to think of proteins. And if you think of proteins, you're going to think of amino acids. Nucleotides, remember, make up DNA and RNA. So we know we can eliminate that choice. Monosaccharides are your carbohydrates. So that is the monomer of carbohydrates. Big carbohydrates know glucose, fructose, and sucrose. Also chitin, which makes up cell wall of fungal cells. But for this question, they're asking what those pumps are made of, and that would be proteins, and the monomers of a protein are amino acids, for number 6. What is the primary function of the cell membrane? Remember, cell membrane can be found in all cells, and it surrounds the cell. Cell membrane is found in all cells and separates the inside from the outside. So it separates this intracellular portion from this extracellular portion. And the cell membrane is found in both prokaryotes and eukaryotes. So it's found in every type of cell, whether you're a eukaryote or a prokaryote, and it separates the inside from the outside. And the primary function of the cell membrane, when we're looking at these answer choices, doesn't determine genetic traits. That would be your DNA and RNA. So we can eliminate that one. Keep this one in mind, defending against foreign particles, because technically separating the inside of the cell from the outside of the cell would defend you against foreign particles. Breaking down proteins for energy, that doesn't make sense, because it doesn't break down proteins for energy. Generating energy from mineral nutrients, that one doesn't make sense either. So our best answer choice here would be B, because the cell membrane separates the intracellular matrix from the extracellular matrix, separates what's outside of the cell from what's inside of the cell. So we would choose B, defending against foreign particles. Number 8, why does the study of cell membranes lead to a better understanding of the cell function? All cell functions occur in the cell membrane. All energy transfers occur at the cell membrane. All cell membranes contain the information for making proteins. All materials needed for cell functions must pass through the cell membrane. So, this one, on the other hand, you're going to want to look at what we can eliminate. So all energy transfers occur at the cell membrane. We can get rid of that one, because that's not true. All cell membranes contain the information for making proteins. That's not true. We know that. That would be found in the nucleus, which stores DNA. All materials needed for cell functions must pass through the cell membrane. That is true. So we can keep this one in mind. All cell functions occur in the cell membrane. That's technically, I could see why you would choose that one, but there technically are these things known as receptor proteins on the outside of the cell. And there's things that can bind to these receptor proteins and cause intracellular responses. So not all cellular functions occur inside of the cell. There are things present on the outside of the cell membrane that still provide cellular function. So this would be a tricky question, but the correct answer you would go with would be D, because all the materials the cell needs to function have to first cross that cell membrane before they can be used. That's our best answer choice here. So number 9, the scientific theory of evolution is supported by different types of evidence. The diagrams below show the skeletons of 2 different animal species. So we have a whale and we have an alligator. How does comparing the skeletons of these animals provide support for the scientific theory of evolution? Now, we can look at these organisms and look at their skeletal features and how they've evolved over a period of time. And we can see that they're sharing the same anatomical structures. So they're both vertebrates, they both have hind legs, they both have ribs, they both have ulna, they both have phalanges, they both have a radius, they both have humerus, meaning that these organisms evolved due to divergent evolution. And we know they evolved due to divergent evolution because they have very similar structures, anatomical structures. And since they have similar anatomical structures, but these anatomical structures don't serve the same function, we can say they have homologous traits / characteristics. And we can prove this because the whale technically has a forelimb just like the alligator. And they both have the same anatomical structures: an ulna, radius, phalanges, humerus. Ulna, phalanges, radius, and humerus. So they're both anatomically similar, but they serve different purposes, meaning that these are homologous characteristics. If they're anatomically different but serve the same function, we would call those analogous characteristics slash traits. So since they have homologous traits slash characteristics, that means they developed these anatomical structures due to a common ancestor. So they would have had to have developed similar anatomical structures due to a common ancestor. And how does comparing the skeletons of these animals provide support for the scientific theory of evolution? It shows possible common ancestry between the organisms, and that's because they have these homologous traits slash characteristics, these traits that are anatomically similar, but their function is different, meaning that they would have had to have inherited that anatomical structure from a common ancestor. So, moving on. The diagram below shows the relationship between photosynthesis and cellular respiration in the organelles in which they occur. Give me 1 second, guys. My cat's here. I just gotta move him real quick. Go play with your toy somewhere else, buddy. Come on. Tabasco. Tabasco. No. Hey. Buddy, no. Come on. Away from the sink. Here. Play with this. Sorry, guys. Okay, I'm back. So, where do we leave off? Which statement describes how photosynthesis and cellular respiration are interrelated? My bad. So, we know that photosynthesis occurs in plant cells, some bacteria, and some protists. In photosynthesis, we need to know this chemical formula is H2O plus carbon dioxide is catalyzed by light to go to C6H12O6 plus oxygen. And the funny thing about this chemical formula is that cellular respiration, which relies on glycolysis, pyruvate decarboxylation, the Krebs cycle, and the electron transport chain, is the opposite of this, excluding light. So when we're talking about cellular respiration, this is photosynthesis. And when we're talking about cellular respiration, which is what you and I undergo to obtain our energy, we are not autotrophic. Autotrophs make their own food utilizing light energy. And we are heterotrophic, meaning that we actually, photosynthetic autotrophs utilize light energy. Autotrophs, the main definition of autotrophs is they just make their own food. We are heterotrophs, meaning that we have to obtain our food / nutrients from an external environment. We can't make our own food / nutrients. So we undergo something known as cellular respiration, meaning that we obtain our glucose from an external environment, which is C6H12O6 plus oxygen, O2, gets broken down into carbon dioxide plus water plus ATP. And if you'll take a look at these two biochemical pathways, you'll notice that they're basically just the opposite of one another. So glucose is a reactant in cellular respiration. Oxygen is a reactant in cellular respiration. Glucose is a product in photosynthesis, and oxygen is a product in photosynthesis. So, which statement describes how photosynthesis and cellular respiration are interrelated is carbon dioxide and water released by cellular respiration are used in photosynthesis, which is true. So it would be B. Photosynthesis releases the energy that is stored during the process of cellular respiration. That is not true. Oxygen is produced during cellular respiration and stored during photosynthesis. Remember, oxygen isn't made in cellular respiration. It's used as a reactant, so that doesn't make sense. Glucose is used during cellular respiration to produce food that's broken down during photosynthesis. Glucose is not converted into glucose in cellular respiration, so that doesn't make sense either. So once again, it's B. Carbon dioxide and water released by cellular respiration are used in photosynthesis. And the opposite for photosynthesis is true. Carbon dioxide and the oxygen produced by photosynthesis are used in cellular respiration. So the products of cellular respiration are used in photosynthesis, and the products of photosynthesis are used in cellular respiration. Number 11, as food travels through the digestive system, it is exposed to a variety of pH levels. The stomach has a pH of 2 due to the presence of hydrochloric acid, HCl, and the small intestine has a pH ranging from 7 to 9. HCl converts pepsinogen into pepsin, an enzyme that digests proteins in the stomach. Which of the following most likely happens to pepsin as it enters the small intestine? So, HCl is what converts, you have to read here. So HCl activates pepsinogen. And in order for pepsinogen to actually break down the proteins, it has to be activated into pepsin. And HCl is a strong acid. It actually has a pH around -7, I believe. So it's a very strong acid. Remember, low pHs are acidic, high pHs are basic, and a pH 7 we consider neutral, biologically neutral. So that means that pepsinogen needs a low pH in order to be broken down into pepsin, or cleaved into pepsin in order to break down proteins. But if you read here, the small intestine has a pH ranging from 7 to 9. If the small intestine has a pH ranging from 7 to 9, and the pH of the stomach, where there's HCl, is 2, that means that pepsinogen is now going from a low pH to a high pH. So if it goes from a low pH to a high pH, it's going to become inactive, because enzymes are pH sensitive, and we need to remember that. They are only active at specific pHs. It begins to replicate. That doesn't make any sense, so we can eliminate that one. Its shape changes to engulf large proteins. That doesn't make sense, so we can eliminate that one. Its activity increases to digest more proteins. We already know that it's more active at low pHs, so it would actually become less active at a higher pH. So we can eliminate that one. And remember, enzymes are proteins that catalyze biochemical reactions. So proteins that catalyze reactions. Number 12, genes for medically important proteins can be cloned and inserted into bacteria, as shown in the diagram below. We have a plasmid vector. Remember, a plasmid is just a circular piece of DNA. Plus a DNA fragment with human genes. We then incorporate this human gene into the plasmid, and we then incorporate this plasmid into a bacterial cell, transforming the bacterial cell, because then the bacterial DNA will actually then incorporate this human gene into itself along with that plasmid. So the genes present in this plasmid will then be incorporated into the overall bacterial genome and can actually be replicated and translated. And then what happens is it gets replicated, and then we break apart the bacterial cell via lysis, and we collect these gene fragments right here. Why can bacteria recognize a human gene and then produce a human protein? The reason being, let's take a look at what our answer choices are. DNA replication in bacteria and humans is the same. That is not correct. DNA replication in bacteria is simultaneous, meaning that they transcribe and translate at the exact same time. Bacterial cells contain the same organelles as human cells. We know that's not true because bacterial cells are prokaryotes, meaning they don't have membrane-bound organelles, and human cells are eukaryotes, meaning they have all those fancy membrane-bound organelles such as the mitochondria, nucleus, Golgi apparatus, smooth endoplasmic reticulum, rough endoplasmic reticulum, all that good stuff. The basic components of DNA are the same in humans and bacteria. That is true. So DNA is, the basic backbone of DNA is always going to be the same, whether you're a prokaryote or a eukaryote. And the basic components of DNA is going to be DNA contains a phosphate backbone, a nitrogenous base, what nitrogenous base is, and also a ribose sugar, deoxyribose sugar, my bad. Should be more specific here. And no matter what type of DNA we're talking about, the basic component of the DNA will always consist of these three functional groups: a phosphate backbone, nitrogenous bases, and deoxyribose sugars. Now, these nitrogenous bases may be in different orders that leads to the translation of different amino acids, but there's still going to be nitrogenous bases. The only thing that's going to differ between different species, different organisms, is the actual orientation and arrangement of these nitrogenous bases that will lead to the translation of different amino acids. But the basic components of DNA are still going to be the same for humans and bacteria. Let's just take a look at D real quick. Bacterial cells and human cells contain the same kind of chromosomes. That is not true. We do not contain the same type of chromosomes. So once again, some of these questions are just common sense, eliminating what's wrong and what's right. But I hope this was helpful. Hope you took some notes. Hope you learned some things, and you guys have a wonderful rest of your night.